Today's lecture is on the structure, organization of DNA, gene and genome. So what we want to learn today is basically what exactly is DNA and what's the difference really between DNA and RNA, and also what are the differences between eukaryotic versus prokaryotic DNA, and in eukaryotes what the differences are between nuclear versus mitochondrial DNA. And once we know all that and the basics of what DNA is, then we want to know about what exactly is the structural composition of DNA and how does one form a nucleotide chain, which will then form a double-stranded helix. And then once we know about the structure of DNA, we want to know about how does DNA get organized into compacted chromosomes. And once we know that, we will then clarify the difference between what a DNA is and how does it differ from a gene and a genome. And then we're going to be learning about the parts of a gene and also the clinical implications of knowledge on DNA structure. So let's move on to the first section of the lecture. So what exactly is DNA? Now DNA is a type of nucleic acid. So nucleic acid in the cell is basically, the main ones are RNA and DNA. And a nucleic acid itself is a macromolecule. A macromolecule is basically a long polymer composed of smaller units. So in proteins, we know that proteins are composed of smaller units of amino acids. Here, a DNA is a nucleic acid, which is composed of smaller units of nucleotides. So if you imagine a Lego piece, the DNA is a finished Lego building and it's composed of small units of Lego nucleotides. So that's how you would put it in an analogy. And then each nucleotide here is composed of a sugar, phosphate, and a base. So this is one Lego piece, two Lego piece, three Lego piece. So each piece has a sugar, phosphate, and a base. That is a one nucleotide, and these nucleotides will string together to form a nucleic acid. Once again, there are two kinds of nucleic acid in the cell, which is ribonucleic acid and deoxyribonucleic acid. And there are several key differences between these two functionally and structurally, but here we will be talking about what the structural difference is between RNA and DNA. So an RNA is a single-strand nucleotide, whereas DNA is a double-stranded nucleotide. We'll look into this later in more detail. And then the other structural difference is that in RNA, the sugar, so this section, the sugar of RNA is made of ribose, whereas in DNA, the sugar is called a deoxyribose. And the third structural difference is on the base composition. So this is the base. And in RNA, the base is composed of AGCU, or adenine, guanine, cytosine, and uracil. Whereas in DNA, this is the same, except for the uracil is replaced by thymine. So in DNA, we don't see uracil, we see thymine instead. This is a little picture that can hopefully help you memorize what the differences are between RNA and DNA. So let's have a look at Dragon Mother's hairdo here. We see that she's got an RNA on her right side. This is a single strand. She's got a single strand of RNA. And on her left side, she's got a DNA. So this is a double strand. This is just a little fun picture for you all to remember what the difference is between RNA and DNA. And here we can see the base and sugar composition. So DNA strands are made of deoxyribose in dark blue, and RNA strands are made of ribose in light blue here. And then as we can see, there are differences in the base. In RNA, we have uracil. This is the orange uracil, whereas in DNA, there is no uracil. We have thymine instead. So once we know about the differences between DNA and RNA, let's have a look deeper into DNA, because DNA is basically the main topic of today's lecture. So DNA is so interesting because it is the molecular instruction of life for all organisms. And organisms here not just implicates us humans, which we are eukaryotic, right? But it also implicates prokaryotes. So prokaryotic and eukaryotic DNA are both very important, and it's the molecular instruction for both types of organisms. Now, there are several differences between DNA in these two organisms, and we'll have a little review. So prokaryotic DNA is basically much simpler than eukaryotic DNA. If you look at a prokaryote structure, they have DNA here. This scribbly little mess is actually DNA, and it's located in the cytoplasm of prokaryotes. Now, whereas in eukaryotic DNA, everything is a bit more complex. First off, unlike prokaryotic DNA, who only have it in the cytoplasm, we have both nuclear DNA and also mitochondrial DNA. So prokaryotes don't have nuclear DNA because they don't have a nucleus, but we do, eukaryotes do, and the nuclear DNA here is what makes 99% of the DNA is all packed into the nucleus, and the remaining 1% is a mitochondrial DNA. So what are the differences then between the mitochondrial and the eukaryotic DNA? So this is a picture of eukaryotic DNA, and this is a picture of mitochondrial DNA. So the nuclear DNA is actually packed into chromosomes. So these, you may have seen a picture of chromosomes before. There are 23 chromosomes in humans, 22 somatic and 1 sex chromosome. And what we basically have is 3 billion base pairs of nuclear DNA, and it is all packed into 23 chromosomes, which is then packed into a nucleus. And this encodes for a lot of genes, 20,000 to 25,000 genes, and it's responsible for everything in your body, whether it is the protein in your blood or the protein pigment in your eyes or the protein pigment in your hormones, everything. So that's what the 20,000 to 25,000 genes are for. Whereas in mitochondrial DNA, it's a bit more simple. They only encode for 37 genes, and that's all involved in the respiratory apparatus. So everything, all the proteins that the mitochondrial DNA genome code for are all involved in cellular respiration, such as NADH. So they're involved in making enzymes such as NADH dehydrogenase. They're involved in making cytochrome B, cytochrome oxidase, which are all respiratory apparatus. So if you learn about bioenergetics, then you'll learn about cellular respiration, and all these enzymes and proteins are coded by the mitochondrial DNA. So only 37 genes. And another key difference is that in nuclear DNA, the genome is inherited from both parents, so that's why they are in pairs. One of them is a maternal DNA, and the second one is paternally inherited and maternally inherited. Whereas in mitochondrial DNA, we inherit solely from the maternal egg, so there's no involvement of the paternal side here. Right. So once we know what a DNA is and what the different kinds of DNA, now we're going to look at the structure of DNA. And to do this, we're going to do this systematically. First, we'll learn about what makes a nucleotide, and then we'll learn about how does a nucleotide make a nucleic acid chain, and in DNA, how does the double helix form. Let's go into the first section, which is what a nucleotide is made of. So each nucleotide in DNA, as you remember, is composed of 3 things. So we've got a sugar, a base, and a phosphate. The sugar in DNA is a deoxyribose. Here is a picture of a deoxyribose. So the deoxyribose is different to the ribose in RNA. Particularly, pay attention to the second carbon. So on carbon number 2 in DNA, we don't have the hydroxyl group that's in RNA. So RNA carbon 2, the ribose has a hydroxyl group, that's OH here, hydroxyl. And in DNA, we only have a hydrogen atom. So that's the difference between a deoxyribose and a ribose. Now, what I want to sort of emphasize here is carbon 5 and carbon 3. The reason for this is we're going to talk about it more later. So pay attention to these 2 positions. Now, the second part of the DNA here is the base, right? The nucleobase or the nitrogen base. So in DNA, the purines are adenine and guanine, whereas the pyrimidines are cytosine and thymine. So A, G, C, T. Those are the bases that forms a DNA. And when we write the DNA sequence down, we only use the first words of each nucleotide. Now, the last part of the DNA is the phosphate group here. So the phosphate is responsible for linking nucleotides together. It can either bind to the 3' end or the 5' end of a deoxyribose. So that's why I wanted you to have a look closer at these two locations on deoxyribose. Okay. Now that we know that a nucleotide is made out of a sugar, base, and phosphate, the next question is, how does a nucleotide form a chain? So what we can see here is that one nucleotide and another nucleotide will form a chain using something that is called a phosphodiester bond. So using the help of an enzyme called ligase, we can form a phosphodiester bond between one nucleotide and a second nucleotide. So the phosphodiester bond links between the carbon 3 of nucleotide, the first nucleotide, with carbon 5 of another nucleotide. So as you can see, this is a string of nucleotide, a nucleic acid chain. And the nucleic acid chain, as you see here, is connected by phosphodiester bonds between the phosphate and deoxyribose sugar, either on the 5' end or on the 3' end of the carbon. So carbon 5, carbon 3, carbon 5, carbon 3, and then there's a phosphate linking them. And here on this side here, so this is the backbone, of the DNA. This is what faces the outside of the environment, and then this is the inside of the DNA. And here we've got the nucleotides. So it's either an adenine, it can be a cytosine, or it could be a guanine or a thymine, but it's all in the inside. And this is the genetic code. So the nucleotide is what forms the genetic code, whereas the phosphate is what forms the phosphodiester bond together with the sugar. Now, at the very end of a nucleic acid chain, we have a free phosphate group at carbon 5. And then in the very end of this side, we have a free hydroxyl group at carbon 3. So that's why DNA is read from 5 to 3, because this is where that's where the free end of carbon is, at the 5 in the start and 3 at the end. Now, we know how a nucleotide is formed, and we know how a nucleic acid chain is formed. Now the next question, how do you connect one chain to another chain in order to make the double helix? So a double helix is formed from a double strand of a nucleic acid chain, and this is formed by pairing the nucleotide bases. So if you remember, we've got adenine, cytosine, guanine, and thymine. What happens is that they connect together. So here we've got the white, these are the nucleotides here. So the red is adenine and the white is thymine, and they are connected by 3 hydrogen bonds. And then we have guanine and cytosine, which are connected by 2 hydrogen bonds. So the bonds between one nucleotide and another nucleotide is called a hydrogen bond, and this is what makes the double strand. So one chain and another chain is connected by hydrogen bonds between the nucleotides. Now, in order to remember the tendency of connection, then we have a little mnemonic here. So to help you remember which nucleotide pairs with what nucleotide. So just remember apples in the tree, adenine pairs with thymine. Yeah, adenine and thymine, apples in the tree. And we have a car in the garage, which is cytosine and guanine. Here we have cytosine and guanine with 1, 2, 3 hydrogen bonds, and adenine and thymine with 1, 2 hydrogen bonds. So if you can't remember which nucleotide pairs with what nucleotide. Just remember, apples in the tree, adenine with thymine, and cars in the garage, cytosine and guanine. Now, the chain length here is indicated by number of base pairs. So if you recall from the previous slide, we talked about how nuclear DNA is made of 3 billion base pairs, which this is 1 base pair, 2 base pair, 3 base pair, 4 base pair, 5 base pairs. So you keep counting until 3 billion base pairs if you wish. And then the specific base pairs allow DNA strands to be complementary with each other. What does this mean? Is that if we know what the sequence of this chain, then we'll know what the sequence is to the other chain because they are complementary to each other. We know that if this is adenine, then the other side must be thymine. So you only really need to know about one strand information in order to know what the other strand is because they are complementary. And another thing is that they run in opposite directions to each other. So as we see, the primary nucleic acid chain here runs from 5' to 3'. But the other chain, which is anti-parallel to this first chain, runs from 5' to 3'. So that's the second characteristic. Now, the inner core, the third characteristic is that the inner core here is hydrophobic and the outside part is hydrophilic. Hydrophobic means it does not like water, and hydrophilic means it likes water. It's got the phosphate backbone here. So that's the 3 characteristics of the double helix. First is that the 2 chains are complementary to each other. Second is that they run anti-parallel to each other. And the last is that the inner part is hydrophobic while the outer part is hydrophilic. Okay, so to summarize the DNA structure, we know that DNA is made out of nucleotides, where each nucleotide is composed of the deoxy sugar and phosphate and nucleobase. And then these nucleotides will make a string of nucleotides called nucleic acid chain, in which one nucleotide is connected to another nucleotide using a phosphodiester bond between the phosphate and the sugar, either at the 3' end or the 5' end. Now we know next that one strand of nucleic acid will bind to another strand of nucleic acid to form the double helix using a hydrogen bond between the nucleobases. Now that we have that out of the way, let's talk about how do we organize this 3 billion base pair of nucleotide into one tiny cell. So if you stretch out your DNA, it will be over 1.5 meters long. And how do you fit this in a tiny 6 micron cell is a very interesting question indeed. And the answer lies in how it is organized and compacted into chromosomes. So here we have the very long 3 billion base pair DNA. And the first thing we do with this DNA in order to fit it into a chromosome and a nucleus is to form a nucleosome. This is a nucleosome. So the DNA first forms a nucleosome. How do we form a nucleosome? Well, the DNA has to wrap itself around 8 histone molecules. So we have 8 histone molecules here, and the DNA wraps itself around the histone molecules to make one nucleosome. Now the nucleosome will string together like pearl necklace, and it will make what we know is a coil called solenoid. So once we have a long string of pearl necklace from nucleosomes, it will coil together to make a solenoid. Now the solenoid will then turn into supercoils that condenses into a chromatin fiber. So this is the chromatin fiber, and the chromatin fiber will coil into a chromosome. Now the Y is chromosome shaped like this. This is one end of the chromatin fiber. We inherit it from our mothers, and another end of the chromatin fiber we inherit from our father. So one fiber from our mum and one fiber from our father, and this is connected with a centromere. So it's called, that's why we say each pair of chromosome is homologous to each other, because they contain genes for similar traits. So you've got the blue eye gene from your mum and the brown eye gene from your father, but they are both genes that code for eye color, so that's why we say they are homologous to another. Okay, so that's how DNA is formed, is compacted into chromosome. Now, another thing that you need to know, another important structure, is a telomere. A telomere is basically the very end of a chromosome. So at the end of the chromosome, we can see a cap at the 3' ends of each eukaryotic chromosome, and this is called a telomere. And we have a look at the sequence of telomere. They are composed of repeats of this sequence: thymine, thymine, adenine, guanine, guanine, guanine. So that's what's in a telomere. And basically the function of telomere is for cell viability and to protect the ends of these chromosomes. The interesting is that with telomeres is that it's been implicated in a lot of anti-aging research, because the thing is telomeric DNA becomes shorter with each cell division, and this has been linked to the development of cellular senescence or cellular aging. Okay, now the big question: DNA, gene or genome? What is the difference between these three terms? So in order to understand, let's go back to a picture of the DNA. So this is a DNA. A DNA is just a sequence, and it contains a coding region in red and a non-coding region. What does that mean? Well, the coding region in red is what will turn into a protein. And that's the important part. That's what we really want to know about, because protein is the function. It carries out function, and it's got jobs to do in our body. So this part of the DNA in red is what makes a protein. It's the one responsible for creating or coding for a protein, whereas the blue parts are non-coding regions. But both red and blue types are all DNA. Now, the coding area is what we call a gene. So gene are unique parts of DNA that code for proteins. Now, the whole DNA is compacted into chromosomes, as you've already known. And all the chromosomes in your body is called the genome. So the whole thing is called our genome. All the genetic information of an organism is called a genome, in which we have 20 to 25,000 protein coding genes spread over the 23 chromosome pairs. Now, it might be a little confusing at first, so I have a little analogy for everyone. So, just to help you understand what the difference is between DNA, gene, and genome. So imagine there's a library, right? And in this library, there is a very, very big cookbook or a recipe book. This recipe book is so big, it contains 20,000 recipes to make cakes. 20,000 cakes. Now, the thing is, because it's a very big recipe book, you need to divide it into volumes. So we have volume 1, volume 2, volume 3, and so on. How many volumes we have? We have 23 volumes. So this is analogous to the genome and the chromosome. So we've got a big genome, which is the whole recipe book. And this recipe book is divided, or the genome is divided into 23 chromosomes. Because it's so big, we have to divide it into 23 volumes equal to 23 chromosomes. And each chromosome, meaning each volume, contains recipes for thousands of genes. We have thousands of recipes in each chromosome. So we can say that the one recipe is one gene. So each chromosome contains thousands of genes, in which each gene will make one protein, or one recipe will make one cake. And the whole cookbook is written in the DNA alphabet. So this is a little analogy just for you to remember exactly what is the difference between a DNA, gene, and a genome. Now, once we know what a gene is, what a genome is, and what DNA is, we're going to have a look at the parts of a gene. So this is the gene here. And if you look closer to what a gene is, we know that it's got a regulatory region and a transcribe region. So a regulatory region is not going to be turned into RNA and turned into a protein, but the transcribe region will. So the regulatory region here contains a part in which the RNA polymerase will bind to, and this is what you're going to learn in central dogma. But here is the transcribe region, or open reading frame, and this itself is divided again into something called exons and introns. So the exons are in red and the introns are gray. So as you can see, the exons here are going to be turned into an mRNA, and this is what's going to be creating the protein. So the introns here, the gray parts, will not be involved in making the protein. So they will be spliced out and they will be removed after transcription. So exons are parts of the genes that will be expressed in the protein. So once again, a gene, this is a gene, is composed of a regulatory region and a transcribed region. A regulatory region is not going to be turned into an RNA, but the transcribed region will. And the transcribed region itself is composed of exons and introns. But the parts of the gene that will be turned into a protein is called exons. Okay, and introns will be removed and are not expressed inside a protein. Right, so the last question here in this lecture is, why do we need to know all this? What's so important about knowing about DNA structure, and what's the importance of knowing about genes and genomes? Well, the reason for why we need to know is because almost every single disease in the human body has some basis in our genes. And by understanding how DNA works, by understanding our genetics and genomics, then we can actually better understand diseases and also learn how to better detect it, how to better treat it, and how to better avoid it. And for example, we have known that there are diseases linked to one gene. So one defect, a defect in one gene can cause a disease. This is a single gene association, such as sickle cell anemia, for example. And it's all based in simple inheritance. But we also have diseases that in which the mechanism of causing the disease is multigenetic, or there is multiple genes that are problematic to create this particular disease, and that's cancer or diabetes or cardiovascular diseases. And there's something called the Human Genome Project, established in around the year 2003, and it's been very helpful in helping us all understand the pathomechanisms of these of these diseases. And basically what this project has produced is a large set of data which can explore multigenetic factors in complex diseases. So by understanding our genome, we can then improve our diagnostics, or how do we detect this disease, and we can improve our therapy, how do we treat it, and also we can tailor or modify our therapy in accordance to everyone's genetic profile. And this is the future of medicine. As we know, it's called precision or personalized medicine. This little picture here, which I copied from the internet, is actually very helpful for us to understand what exactly is precision medicine, or the future of medicine. As you can see, this is a population, for example, a population of Indonesian people, and among this, we all look the same, we all look very similar, and all this time we're all prescribed the same medicine for the same disease. But apparently, we are not that similar to each other. And based on our DNA profiles, we can actually divide the population into people who will respond very well to a particular drug, people who will not respond to a particular drug, and people who will have very bad side effects if they take the same drug. So one medicine or one drug can have very different effects on different people, and that is all detectable by our DNA profile. So that's the future of therapy, as we know this as precision medicine. Now, a little bit more about the Human Genome Project and how do we do it. So as you can see, this is a population of people, and among this population we want to find DNA sequences, so single nucleotide polymorphisms, which are DNA differing in a few base pairs. So we've got a single nucleotide polymorphism here, and we detect it in this population and this population. On this side we have the people with the disease, and on this side we have people without the disease, and we'll detect varying SNPs on all these populations and in both groups. And if we find that there's the same amount of SNPs or single nucleotide polymorphisms between the two populations, it means that this polymorphism is not associated with the disease. Same with this one, we can see the number of dots or polymorphism here is the same in these two groups, so no association to disease. But have a look at polymorphism 3 or SNP3, we can see that there is much more, the prevalence is higher here in the people with disease compared to people without disease. So by this we can tell that when you have this particular polymorphism in your gene, if your DNA has this particular difference, then you can say that is the root cause of the disease because it is associated with the disease. So that's one of the ways we can pinpoint what parts of our gene is implicated in what disease. Alright, so now that we've learned about the DNA structure, function and organization, then I think that concludes the end of the lecture. Thank you for listening.